In this topic, we're going to discuss the link reaction and Krebs cycle. So by the end of this topic, you should be able to outline the link reaction and outline the Krebs cycle. So just to recap, what are the 4 stages of respiration? You've got glycolysis, link reaction, Krebs cycle, oxidative phosphorylation. So link reaction, Krebs cycle, oxidative phosphorylation only occur if oxygen is present. If oxygen is not present, you have anaerobic respiration occurring. So what happens if oxygen is not present? Well, the pyruvate is going to stay in the cytoplasm, and it's going to be converted into either ethanol, which is in plants, or lactate in animals. So it undergoes anaerobic respiration. If oxygen is present, the pyruvate will enter the matrix of the mitochondria by active transport. Now, this pyruvate does possess potential energy that can only be released using oxygen in a process called the Krebs cycle. Before it enters the Krebs cycle, these pyruvate molecules must first be oxidized, and we oxidize it in a process called the link reaction. So what is the link reaction? You have pyruvate being converted into acetyl coenzyme A. So first of all, it's decarboxylated, where carbon dioxide molecule is removed, and then it's dehydrogenated, where hydrogen's removed. Where does that hydrogen go? The NAD will pick up the hydrogen, and so the NAD becomes reduced. Then the acetyl group combines with coenzyme A to give acetyl coenzyme A. So the acetyl group is a 2-carbon molecule, and it's going to combine with a cofactor called coenzyme A. The acetyl coenzyme A is then going to enter the Krebs cycle. So what is the overall reaction? You've got pyruvate plus NAD plus coenzyme A forming acetyl coenzyme A plus reduced NAD plus carbon dioxide. So if you were to write 2 columns, one for ATP, one for reduced NAD, so that you can tally up what the overall yield is, you'll notice in the link reaction you have no ATP being formed, but you do have 1 NAD reduced being formed. Remember that for every glucose molecule, you have 2 pyruvate molecules being formed. So 2 pyruvate molecules will be coming into this reaction, so we times it by 2. So overall, after the link reaction and the glycolysis reaction, you've got 2 ATP being formed and 4 reduced NAD. So keep this in mind because you need to keep tallying as we go along. So why is acetyl coenzyme A important? Coenzyme A is made up of vitamin B. It's got an organic base and a sugar. It carries the acetyl group from pyruvate into the Krebs cycle. The acetyl coenzyme A molecule is very important because most molecules that are used by living organisms for energy are made into acetyl coenzyme A first before they enter the Krebs cycle. Most carbohydrates and fatty acids can be metabolized into acetyl coenzyme A to release energy. In the case of fats, these are first hydrolyzed into fatty acids and glycerol, and then the fatty acids are eventually turned into acetyl coenzyme A. Interestingly, acetyl coenzyme A is also involved in changing excess carbohydrates into fats. Let's have a look at the Krebs cycle. The Krebs cycle was named after Hans Krebs, who worked on the sequence in 1937. Where does it occur? It occurs in the matrix of the mitochondria, and it involves a series of very small enzyme catalyzed steps. So let's go through them. The 2-carbon acetyl coenzyme A from the link reaction is going to combine with a 4-carbon compound called oxaloacetate, and it's going to form a 6-carbon compound called citrate. So it's quite easy to remember. 4 plus 2 makes 6. The first step is to count the number of carbons. You can see that a 4-carbon molecule combines with a 2-carbon molecule to produce a 6-carbon molecule called citrate. It then forms a 6-carbon intermediate, 5-carbon, 4-carbon, and then eventually your oxaloacetate is going to be regenerated. Notice how when it goes from 6 carbon to 5 carbon, 5 carbon to 4 carbon, carbon dioxide is being removed. And remember that this happens twice for each glucose molecule. Let's now have a look at the electron carriers. You can see reduced NAD and reduced FAD. So where do these occur? We've drawn it as a little piggy bank because it's an electron carrier. Okay, so NAD how many NAD are there? You've got 1, 2, 3. Because this occurs twice, you've got 6 NAD, reduced NAD being formed. How many FAD reduced are formed? 2. So let's go through this step by step. First of all, you have the 6 carbon being decarboxylated and dehydrogenated to give a 5 carbon compound. Then this 5 carbon compound is going to be decarboxylated, dehydrogenated to form a 4 carbon molecule. This will undergo a series of steps where you have further decarboxylation and dehydrogenation to eventually form oxaloacetate again. So you have electron carriers called NAD being reduced, and then 1 molecule of ATP is formed here. So remember to draw your columns on each side to count the number of reduced NAD, FAD, and ATP being formed. How many do you get? So press pause and write them down and then check with my answer. Did you get 6 reduced NAD being formed, 2 ATP, and 2 reduced FAD? Remember to add them up to the previous reaction, so you should have 10 in total, reduced NAD in total, 4 ATP, and 2 reduced FAD. So what is the overall Krebs cycle? You've got citrate being formed from acetyl coenzyme A and oxaloacetate. The citrate is then decarboxylated to give carbon dioxide. It's dehydrogenated to release hydrogen ions, which are picked up by NAD and FAD to reduce them. And then oxaloacetate is regenerated. And you have 1 ATP being formed by phosphorylation. Remember that the Krebs cycle will go around twice for each molecule of glucose. So the overall products you have are 4 carbon dioxide, 2 ATP, 2 reduced FAD, 6 reduced NAD. So this is a table that you can use to check your products along the way. So copy out this table and test yourself. Glycolysis, you should have 2 ATP, 2 reduced NAD. Link reaction, you'll have 2 reduced NAD, 2 carbon dioxide. Krebs cycle, you should have 2 ATP, 6 reduced NAD, 2 FAD, and 4 carbon dioxide. So your total will give you 4, 10, 2, and 6. Where are these going to be used? Well, the ATP is going to be used. The reduced NAD is going to go to the electron transfer chain, as well as the reduced FAD, and then the carbon dioxide diffuses out. So, what is our summary? The link reaction converts pyruvate into acetyl coenzyme A. The reactions involved are decarboxylation and dehydrogenation, and your products are 2 carbon dioxide and 2 reduced NAD. What about the Krebs cycle? Can you explain the Krebs cycle? Acetyl coenzyme A, which is a 2 carbon molecule, combines with oxaloacetate. What does it form? Citrate. This gets decarboxylated and dehydrogenated to form a 5 carbon molecule. A series of steps occur to regenerate oxaloacetate again. So ATP is also formed. So what are your products? You have 4 carbon dioxide, 2 ATP, 2 reduced FAD, and 6 reduced NAD. And that concludes our lesson, the end.